Welcome to the Business of Biotech Summer Executive Sessions. I'm Matt Pillar, Chief Editor at Bioprocess Online, and on today's show, we're talking myeloid cells and transplant surgeries and a few other things with OSE Immunotherapeutics Director and CEO, Alexis Peyroles. OSE is a clinical stage biotechnology company focused on developing and partnering on therapies to control the immune system for immuno-oncology and autoimmune diseases. Alexis joined the company in 2013, bringing along with him more than a decade of global life sciences finance leadership experience with companies like Sanofi and Guerbet. Alexis, welcome to the show.
Thank you very much. Pleased to be here.
Yeah, it's great to have you. We're honored. I want to take some time to get to know you a little bit better before we get into some of the science that you guys are working on and how you're going about that. Your education, I noticed, is in business finance, which it's one of those things that you can kind of take a thousand different directions out of school. And I'm curious why you chose life sciences with your first job at Sanofi.
Well, it's I think it's a mixture of opportunity, choice, as you said, and luck. I mean, I come from a family where my two grandparents were medical doctors, so I heard a lot about the importance of science and drugs and so on, and how to help people. And when I did my business school in France, you had this opportunity to stop between the second and third year and to do 1 year full-time traineeship. So I had the opportunity to join a French pharmaceutical company, which was called at that time Roussel-Uclav, became Roussel, then Sanofi. And I had the opportunity to work with the marketing director, who was at that time Pascal Soriot, who is now the CEO of AstraZeneca. So he was my first boss, and I was very lucky to learn a lot with him on this. So it gave me the willingness and the opportunity to continue my career in the life science for a couple of years and to work abroad in Japan and also in the Baltic states. So that's the reason why I decided to continue like that. So, sometimes you have some opportunities that you have to catch, and then it decides for you the path forward.
Yeah. And it seems like, looking at the timeline of your career, it seems like you had an opportunity to sort of vet whether you were on the right path, because you left life sciences for a little bit, a couple of years, I think. And again, this isn't something that I typically see on the CV of a biopharma CEO, but you left and went to work in the advertising industry of all places. What prompted that move?
Well, I come also from a family where you have a lot of entrepreneurs. So I decided to start something new and to create one of the subsidiary of an advertising company based in Lyon. So it was also a very pragmatic and personal choice, because my girlfriend at that time was based in Lyon, and she became my wife. So it was a very good move also.
Oh, congratulations! Yeah.
So that's the reason. But really, I really wanted quite early on, after leaving Sanofi, working in a very big group, where you learn a lot when you work in a big corporation, but you want to try something new and to try to build structure, to build a company, and that's what I decided to do. And then I would say the life science, I missed life science and the pharma industry and the interest for patients, for example. So that's why I decided to come back and work for Guerbet.
Yeah. So you came back and worked for Guerbet, which was that's a medical imaging company, correct?
They are doing contrast media products, so working in the medical imaging, but these are actual drugs. I mean, you develop them as a medicine.
Gotcha. Okay, good. So you did that for a little bit, and then what prompted the move to get back into biopharma?
Well, I was lucky while working at Guerbet. They had the opportunity to do an executive MBA at Imperial College, where I learned a lot about innovation and again starting new structures and so on. So while we were in Brazil, we decided to come back to France for personal reasons, and that's how I decided to search the best project I could find in a biotech and help build and develop a specific biotech, and I found OSE and I met Dominique Costantini, its founders, and in 2013, and we got along very well. So that's how all this started for me at OSC Immunotherapeutics, and working for cancer and in immuno-oncology was very important for me, because I mean, for family reasons, it was important to come back to try to, in this field, to bring something for the patients and for the families.
Did you, so when you joined OSC, you joined as CEO. Was that your first position with the company?
I joined as a CFO and in charge of business development. Dominique Costantini was the CEO at that time. And we together we did an IPO. We merged with another biotech to focus on research, and we're going to talk about that, but on research in immuno-oncology and in autoimmune disease. And then in 2018, we both, Dominique and myself, thought that it was the right time to switch a bit roles, and that's how I became a CEO, and she became chairman of the board, and kept a very operational role in terms of clinical development.
Excellent. So yeah, let's get into the work that OSE is doing. I touched at a super high level on sort of what your company does, but why don't you give us a little more detail on that? What is it that you guys are seeking to do?
So we are a typical R&D biotech. So we focus mainly on research and clinical development. We try to have innovative targets or innovative type of cells compared to other biotechs to differentiate ourselves a bit. And we have been, we started the company, we were two, Dominique and myself. We are now 45 with especially a strong research team, which is based in Nantes. Nantes is on the western part of France, and it has been a cluster of transplantation and immunotherapy for the past 30 years. So we try to leverage this knowledge in order to find new targets, new drugs, which could be of interest in these two areas of expertise that we have, immuno-oncology and autoimmune disease. And we do also try to get partnerships quite early on with big pharma companies. That's what we did with a French pharma company called Servier and a German company called Boehringer Ingelheim, which helped us continue the development of our product. So we have clearly a business model where we do R&D, and we don't want to commercialize for the moment our own products, for example, and we try to develop our portfolio with various mechanisms of action, various products, which will tackle the resistance that we see, or the unmet need in both immuno-oncology and autoimmune disease.
Mm-hmm. Yeah, it's interesting the way that you paint the picture of the company as R&D, clinical, early stage. Yet, and we'll talk about this a little bit later in the conversation. You've got some pipeline candidates that are pretty far along.
Yeah, well, actually, when we started the company in 2013, our lead asset was already in phase 2, and we were able to buy back this asset, which had been developing in the US. And initially, what we thought would be that we would be able to acquire other clinical assets in immuno-oncology. But then the tsunami of the immuno-oncology arrived, and these kind of assets became much too expensive for a company like us. But on the other hand, we understood that there was still a lot of work which could be done in research, in immuno-oncology and in the autoimmune part. And that's why we decided to focus the company on this and to recruit the right kind of people in order to help us achieve this objective. So we had a change of business model in 2014-15, and since then we have this R&D focus as you described it.
Okay, great. So let's talk about that. Part of that focus is on myeloid cells. Why are these key to harnessing the power of the immune system to fight disease?
Well, I think it's clear that when you look at immuno-oncology and the great progresses that you had in the past 3 to 4 years, we all know that T cells, T lymphocytes are very important to attack the tumor. And on the other hand, in the autoimmune part, you need to block them so that you avoid serious disease. But coming from transplantation, coming from graft, our research team knew well the other types of cells which are important in the immune system. And myeloid cells, for example, they represent 50% of the cells in the immune system. So they are key to help us fight against cancer, for example. So the idea for us was to leverage this knowledge that that we have in transplantation, that we have in the graft area, knowing even before the immuno-oncologist, the immune system and these myeloid cells. And as you have less companies, biotechs, which are focusing on that, it was a very good way for us to differentiate ourselves.
Mm-hmm. So tell me more about where that knowledge comes from. You know, you've alluded to the importance of transplantation surgery as a source for new ideas, for drug targets, for immuno-oncology and autoimmune diseases. That's really intriguing to me. What about this transplantation history has enabled those learnings and that intellectual property to exist at OSE?
Well, in transplantation, it's exactly a mirror compared to cancer. You want to make sure that your own system will not reject the organ that you have implanted. So that's why transplant people have been working on all the immune system, on immune cells for the past 30 to 40 years. And they had successes earlier compared to immuno-oncology. So we know well all these type of cells, and that's how we have this idea. For example, the product that we have licensed to Boehringer Ingelheim, which is targeting macrophages, so it's a type of myeloid cells on SIRP-alpha, on the SIRP-alpha CD47 pathway. It was discovered by our team in the year 2008, 2009, that SIRP-alpha could be found on myeloid cell while working on transplantation. So it was not working in transplantation when you were blocking this type of pathway. So as a contrary, it means that it should be working in cancer. And that's exactly the type of thing that we apply to find our new ideas. We just announced last month in a collaboration with the hospital and a university lab in Nantes that we have a new target called CLEC1. And this new target is also on myeloid cells and also was discovered while working in transplantation. And as you block it, you think that it will be working in cancer. So the idea is how you use this knowledge that you were that we had when we were focusing in transplantation to fight against cancer, and not being on T cells to differentiate and attract pharma companies who want to find ways to combine drugs, to combine treatments, and to have several types of treatment in order to fight this vicious disease.
The Business of Biotech is produced by Bioprocess Online in partnership with Cytiva. If you're the leader of an early to clinical stage biotech, you need to check out the resources that are hand-curated for people like you at cytivalifesciences.com/emerging biotech. The Knowledge Center there is chock full of articles, webinars, videos, podcasts, and other content that's ultra-focused on helping new and emerging biotechs chart the course to the clinic and beyond. Check it out, cytivalifesciences.com/emerging biotech. Are there specific transplant surgeries that have kind of enabled these learnings, or is it pretty universal? I mean, you know, I guess my assumption, and it's purely assumption, would be we're talking about you know, bone marrow, for instance. But is there more to it than that?
No, it's clearly it's more universal than that. I mean, in Nantes, this is one of the hospitals in Europe where they do a lot of kidney transplantation. So a lot of work has been done on kidney transplantation, but you have various models in mice or in primates on bone marrow and so on. So it's you cannot just focus on one type of surgeries or one type of transplantation, which will give you specific targets. But that's quite universal. For example, we have a specific collaboration with another hospital in France, in Lyon, so on the other side of the country, which is well known, called Centre Léon Bérard, and they have access to patients who have cancer, and we have a specific artificial intelligence program with them in order to find additional myeloid cells targets. So you cannot, we don't rely only on transplantation. That's the basis of our knowledge, and that's where we come from. But now we have expanded all this in order to find these new targets, which are of great interest for the clinicians.
Yeah. Yeah, and that's interesting. The AI angle is interesting to me because I wanted to ask you just what the process of gleaning this insight looks like. And it seems like there'd be a lot of trial and error on, like, let's observe what myeloid cells do in certain transplant environments with certain organs, with certain people. That's probably a lot of inputs to arrive at an outcome. Is AI helping to enable those?
Yes, it does actually, more and more. I mean, the way we were doing that 10 years ago is completely different nowadays. You can use, specific algorithm. You do a lot more of gene research. I mean, you can analyze tons and tons of data in order to be quicker in the way you identify potential targets and the area of interest, as you mentioned. So it means that the amount of the data that we have access to it's much higher now than it was before. Nevertheless, you still have, for the moment, some bottlenecks. I mean, you still need to test your product in mice first. You still need to see what it will give in preclinical model and so on. What we develop also in order to address this kind of bottlenecks using artificial intelligence and using 3Ds, we have access now to tumor samples, and we are able to kind of put them in volume in order to test our products. And it's has been developed by someone in our company. I mean, we have a specific patent on that, and it does help us accelerate the identification or the choice between several targets or several products. So there is still a lot of things which could be done in order to be quicker from the ID that you get, I would say, to the product being tested in clinic. And the importance for us is to attract the right talents who can do that, because there are very few people actually who can manage both the biology part and the computer part. So you need to find someone who understands both. And if you have these people, then you will be able to expand the number of projects that could be of interest and develop a process where you have to make the right decision to stop some projects. As you said, it's trial and errors. Not everything works, that's for sure. But you use more and more computational work in order to do that. And for a company like us, who is in a race against other biotechs, but also other big pharma companies who have much more resources than us, it's very important to be always at the forefront of this kind of tools in order to convince them that you have identified the right target, an interesting product, and so on. That's part of our job to be always at the forefront on this point.
Yeah, and you bring up a really interesting point around building a team and ensuring that team has people on it who are capable of both understanding the biology, but also the computational aspect. And you also mentioned earlier that you've grown the company from 2 to 45 employees. How have you gone about the HR, the human resources aspect of bringing people on who kind of can bridge that gap between biology and artificial intelligence? What's the secret there?
I don't know. I played rugby for 10 years, so I know that in a team you need to have, tall guy, fat guy, fast people, and so on. You need a team of various characters and various personalities and abilities to do specific work. But joke aside, first of all, you need to attract them, and you attract them if they see that you do a strong science, if science is important for you. And that's clearly the motto we have at OAC. I mean, we need to strong science to be convinced, strong biology to be convinced that we move forward with one or two specific projects. So that's the way they see that you give them the resources, the freedom also to pursue some specific project. Sometimes we have one specific project. We don't see clear results for 1 or 2 years, but you continue. And after 2 years, you have something which happened, which makes it interesting and worth it in terms of investment, in terms of resources and time. And on the other hand, you clearly need to identify what are the specific points that you are missing, what are the competencies that you are missing, and bring them together in order to make sure that the people you hire will bring these competencies, but will fit well within the team. So that's clearly the two things that we try to have. I mean, you need to have a strong team spirit, and you need to have people who can work well in team, which is not always the case. And sometimes we had to make decision not to hire some very clever people, but thinking that they will not fit well within the team and will bring more disadvantages, I would say, than the positive aspects.
Yeah. As it relates to the R&D culture of your company and that focus, what how did you go about strategically pacing your hires? Like, I guess I would ask, where did you place priority around the titles and positions that you were filling to kind of meet that R&D objective?
Well, we clearly started quite early on the research side. So as the number of projects were increasing, we brought more researchers, PhDs and so on, trying to make sure that we identify the right people who could be project manager or project leader, for example, for this kind of interesting early projects, and then bringing additional researchers to help these people. And then as we moved forward toward the clinic, then we hired more people who knew well how to do clinical development, who had been working in pharma and in other biotechs for the past 15 to 20 years. So that's the way we try to fill the gaps, and it's a never-ending story, actually. I mean, we are going to hire 4 to 5 people until the end of the year, because I'm very lucky to work with people who search and find new targets of interest. So then you want to develop them and bring them towards the clinic, because that's clearly the judgment that you need. I mean, does it work in humans? That's the only interest we have. It's very good. I mean, the science is very important. The understanding of the biology is very important. But you cannot be only mechanistic in your approach to R&D. What you want is a result. You want data, and you want positive results. That's the idea.
Yeah. There's also the, and I want to ask you about this, even though I understand that you're an R&D focused company. As I mentioned earlier, you've got Tedopi. Did I pronounce that correctly?
Yeah, exactly.
As far along as phase 3, and other candidates in the clinic. And of course, that's the ultimate goal, is to get some of these ideas to move forward. As you do that, give me your perception or your perspective or philosophy on how early in that R&D process do you start to determine the viability of these therapies from a manufacturing and potential commercialization standpoint? What challenges do you see there in the actual process of potentially taking these things to market?
We try to do that quite early, actually. For example, we have a new bispecific platform that we are developing, and we started to work on that 2 years ago to address specific resistance to checkpoint inhibitors. And as a backbone for this platform, we choose our own anti-PD-1, and we choose it because the productivity and the stability of this backbone could be good in a bispecific platform, knowing that that's one of the main challenges when you want to do a bispecific on a format. So that's clearly something, and we are learning a lot. I mean, obviously we don't address or start a project now the same way we started when we started a project in 2017, for example. We have learned what is important, how you can gain speed on things, where you need to spend time on other things, and productivity, manufacturing aspects are essential in our business. I mean, you need to make sure you have a nice yield, for example. Otherwise, there's no way you can continue to work on the development of a specific product. So that's why our company is really organized like a dossier you present to the authorities, the FDA or EMA. I mean, you have people who are specialized in biomolecular, you have engineers, you have people from regulatory, from CMC, people from tox also, so that clearly you work as a project team for something which has to become a product which could be registered if the results are positive enough. And that's how we started with Tedopi, as you mentioned. I mean, we had positive step one results in our phase 3 in a unmet need because it's post-checkpoint failure. So that's very important to assemble the right team towards this. And for the newer project, for example, I mentioned on the myeloid cells target CLEC1, it's quite early. I mean, we still have to define which lead candidate we could have, but we are already thinking about what types of models we are going to develop, in which indication we could use it, and how do we make sure that the cell line, for example, will be the most efficient possible based on the issues we might have in the past on other projects.
Mm-hmm. You mentioned that oncology and immunotherapy are near and dear to you, and sort of it's important to you that those are the things that you're working on. Within those kind of broad umbrellas, how do you go about prioritizing which targets you're going to go after, which indications you want to tackle?
Well, that's the most difficult part. That's difficult because when you have several interesting projects, you would like to do everything at the same time, but you have limited resources. So clearly that's the idea here, and that's why it's important to have people who have been involved in clinical development mainly. You really need to make sure that you address unmet needs, and that you are not going to spend 10, 20, 30 million euros to have a treatment which could bring only a small benefit compared to Doliprane or paracetamol, for example. I'm just giving you but sure. Sometimes you have to come back to the basics because you can be so interested by the science, by the biology, that you might forget sometimes that there are some treatments which do work, which are very cheap and so on. So in some specific indications. So that's why you need to come back to which kind of unmet need are we going to fill? Where are we going to develop this specific product on this specific target? And are we going to bring something sufficiently beneficial for the patients so that it makes all the efforts worth them?
Yeah. Yeah. As you go through this process, I wanted to ask if you have encountered any specific process challenges that maybe held up the potential for the yield that you mentioned, and how you went about solving those process challenges.
Yeah, I mean, you for example, I mean, we had some challenges in one of our projects, and the yield was not high enough, so we could have stopped it. But fortunately, I mean, you have we do work in a network of companies who are specialized in solving some issues. And I give you an example. We work with a company called MapSilico, which is specialized in silico data. So they have algorithm and they can help you improve some monoclonal antibodies so that you can solve the yield issue or other issues. And by forging these kind of relationships with these people, you can accelerate the development, tackle some issues, solve some issues. I have also some example where we could not solve the issues, and you have to stop the project. The important thing is to make sure that you still have enough project to refurbish, I would say, your portfolio, and make sure that you continue to have enough product which will be at clinical stage in the future for the development of the company. And we are lucky to have this, that's clear.
Are there any common threads or hallmarks of that stop sign that says, hey, we're not going to be able to do this anymore? Like, is there I guess I'm asking if there's a clear indicator or a common thread among those indicators as the leader of the company that you've seen that you could share with others. Say, look, when it gets to this point, you got to cut bait and move on.
Well, when you have tried on 2, 3, or 4 different kind of models and you don't have any results, even though on the paper the biology seems very interesting and so on, it means that as a company we have not found the right path forward for this target, for this product, which does not mean that some other people might not find it, and we might see in the future other biotechs or pharma companies who will be working on the same pathway but addressing it a bit differently. So we might want to come back to this and say, that was clever, let's try on this way. So that's a possibility. And sometimes also we've got a lot of academic collaborations, and sometimes we get contacted by some academic researchers who want to try our product on something, and we say, why not? I mean, we'll see, but we don't believe in it. And then they come back with very interesting results, and you see, yes, that's the way you move in this world. I mean, the world of R&D is made of trials and errors. We don't pretend that we know everything, far from it. And so you need to have this kind of partnerships, not only with big pharma companies, but also with academic labs and so on. And that's the way you will progress and make sure that you improve your project and your targets.
Yeah. So we're running a little bit short on time here, Alexis. So I want to kind of wrap this up, but I'm curious what the short term and maybe a little bit longer term goals for OSC are. At this point, you've got, like I said, you've got some pretty mature clinical candidates, and you've got this focus on R&D. Is the goal maybe to, go commercial with something to fund more R&D? Is it, what's kind of the next step?
Well, clearly for the next 2 to 3 years, the important thing for us is to bring more clinical stage products forward, to get clinical results, clinical data, because it will validate in a way our model and our abilities to find new interesting targets. Then if we go a bit broader than that, and after 2 to 3 years, my goal would be exactly that. I mean, if you can find a product or a target that you can develop on a specific indication, which would not require millions and millions of euros. I mean, if you address the non-small cell lung cancer market, you've got big pharma players, so you're not going for that. But in the autoimmune disease part, for example, or even in immuno-oncology, you can still find some niche indications. And I come from the pharma world, as you mentioned. I've been marketing drugs before, like most of the team in the clinical development that I have. So we could do that. But first, we need to build the house and show that our model is working. We are able to find innovative targets and to get positive clinical data for that. That's the goal for the next 2 to 3 years.
Excellent. Any concluding thoughts for our audience of new and emerging biotech leaders? Maybe some more references to rugby, like put your head down, carry the ball, and move the pile, right?
Yeah, and never stop, Just be sure that you will face people who want to tackle you, but you have to avoid them or fight them.
Yeah, excellent. Alexis, thank you for joining us. It's been a fun conversation and an honor to have you on the show.
Thank you very much. It's been a pleasure. Bye-bye.
So that's Alexis Peyroles. I'm Matt Pillar, and this is the Business of Biotech. We're produced by Bioprocess Online and sponsored by Cytiva, which offers a host of great resources for emerging biotechs at cytivalifesciences.com/emerging biotech. Check it out. Go to bioprocessonline.com to subscribe to our newsletter. And if you like what you're hearing here, subscribe to the podcast on the pod platform of your choice and give us 5 stars. In the meantime, thanks for listening.